am here from the University of Washington in Seattle. Thank you very much to Unruly for the opportunity to be here and to New Lab for hosting. What my lab does is to develop new materials for additive manufacturing. Right, if I look at the type of materials that people are using now to 3D print, these are materials that were developed 60, 70 years ago. And so what we do is we think about what are the material requirements for 2, 5, or even 30 years from now. And so while I'm a polymer chemist, I'm actually super excited by what synthetic biologists do. Right? Because what they do is they can take a cell, whether it's a mammalian cell, bacteria, or yeast, and they can genetically engineer it such that they take very simple molecular inputs and then transform them into more complex molecules. All right, so this is pretty incredible because as an organic chemist, that's really hard to do. But with bacteria or yeast, you feed these organisms these small molecule precursors, could be CO2, could be methane, sugars, and then they transform them into molecules like ethanol, right? Paclitaxel, insulin, right? So these are molecules with different ranges of complexity. And so in fact, all of the insulin that's produced in the world today comes from yeast or bacteria, large-scale fermentation. All right, so the limitation, though, of synthetic biology is that you need to have a controlled lab environment. Right? And so if we're talking about applications that are in resource-limited environments, so we're talking about military missions in remote areas, agriculture, remote areas on the planet, or even space missions. Right? So some of the challenges that all of these areas have in common is that first, these are remote or isolated settings, so it takes effort to get resources to those areas or to build the equipment that you need. There are also weight and space limits for a lot of these areas as well. So if we're talking about a mission to Mars, a human mission to Mars, that's a 2-year round trip. You have to take everything with you, right? So everything over the course of 2 years. I have a hard time figuring out what I need for 2 days to come out here, right? And so you have to be very careful. You have to be very, very conscious about what you're going to take with you. And ideally, you want to take everything. There are also long-term storage requirements when it comes to working with biologicals. So a lot of times you need to have cold storage and maintain that cold storage over the entire period of time. If you're talking about vaccine deliveries across different areas of Africa, then that's a huge challenge. So there's also environmental exposure related to all of this as well. So whether it's just environmental exposure from the sun, or if we're talking about being able to work with these types of organisms in space, then you're also concerned about microgravity, right? Also radiation from gamma radiation as well. And so those are effects that start to impact the ability of these organisms to produce molecules. And then ideally, we'd like for all of this to happen in the hands of a non-expert user. Right? So there's one thing about having everything in a controlled environment where you've trained that person to use it, but ideally, and in many of these cases, you want a non-expert user to be able to handle these materials and deliver the products that they need. So our concept here is to change the idea of biomanufacturing away from what's traditionally a batch process to a continuous one. So in a batch process, what this means is that you have a large reactor, you combine everything, all the cells, you let it react for a couple of days, you isolate the product. But what if you don't have to break it down at the end of the reaction? What if you can keep it going over extended periods of time? And so the way that some of the advantages of doing this is that means that there'd be reduced reactor downtime. It would simplify product recovery because you can keep the cells trapped within these matrices. You get around some issues of product inhibition with these systems, and then also enables on-demand production. So I'm not going to go through all the details of what we've developed with respect to the materials, but this in the bottom section here just kind of outlines what we thought were some of the requirements. But the one thing I want to highlight here is that the material platform that we've developed is agnostic to the cell type. So we can incorporate yeast, bacteria, algae, all different types of organisms into the materials I'm going to show you. So in this one slide, this is my last slide, I just kind of captured what we've developed and what we're really excited about right now. And that is we have a hydrogel material that's shown in that upper left box there. So what we can do with this hydrogel material, it's kind of like a hair gel, kind of a thicker hair gel. We can incorporate the microorganisms into that gel, but it has a consistency of like a toothpaste, right? And so a toothpaste, if you squeeze that tube, the material flows out, but then it's a nice cylindrical structure. And so that's what we use in our printing process. So we have a controlled extruder that allows us to extrude this gel that contains the organisms inside to create a three-dimensional lattice construct. And then we do a quick light process to freeze the material. And then once we've printed that construct and we've cured it with light, then we can go into this lower left-hand box here, and we can use that as an immobilized catalyst. All right, so we can take this printed structure, and in the case of fermenting glucose to make ethanol, right, we can put that structure into a solution of glucose. The organisms transform that glucose into ethanol. However, this structure is reusable. All right, so once that transformation is done, we can take it out, put it into a fresh solution of glucose, and the organisms keep going. Right? And we can keep this process going now for 1 year. And the only reason why we stopped at 1 year is because my graduate student felt like, okay, we've reached a point, right? Point made. And so while these structures are reusable, and that's really exciting, these also have shelf stability. So in the lower right-hand box, I show you the dehydrated forms of these structures. So if you take these hydrogel structures, you remove the water, you're left behind with just the polymer encapsulating these organisms. It's dry. And that dried construct is shelf stable. And it says 3 months here, but that's a mistake because these structures actually shelf stable for up to 12 months. And at any point in time, you can take that structure, you put it into solution, and the organisms become active again. And they pick up, the productivity is exactly where they left off. So ethanol is a simple example of what we can make, but we've also demonstrated that we can make anti-inflammatory agents, we can make antibiotics, we can make large proteins like seed protein as well. And so there's a whole range of products that can be made and diffuse out of these systems that we can collect. So hopefully what I've shown you is just our concept for what the potential here is for combining polymers with synthetic biology, and I'm happy to have any further discussions with anyone interested.